a pleasure to introduce today's seminar speaker. Chelsea's visiting us from just down the road in Denver area where she works at Charlotte in Clearsight. She's Director of Product Development at Charlotte Technologies, which is in Aurora, where she leads research and development on medical devices that use bio-inspired Charlotte surface texture and control biological adhesion. She got her BS degree in Material Science and Engineering from the University of Florida in 2006, and MS and PhD degrees in Biomedical Engineering from the University of Florida in 2008 and 2010. Her doctoral research focused on developing biomaterials to control adhesion of both marine fouling organisms and mammalian cells. She completed an NIH postdoctoral fellowship at the University of Colorado, Boulder, in the ANSEP research group, where she developed user-controlled, dynamically tunable biomaterials to study clinical stem cell differentiation and the progression of heart disease, valvular, and interstitial syndrome.
Everybody situated. Thanks for the introduction, Seth. I'm really excited to be here today to share with you my work on the development and translation of these engineered biomaterial interfaces for controlling biological adhesion. No?
Of course. Oh, it's a smart board.
Oh, I just have it.
Right.
No?
Try the arrow key on that.
I guess I'll just try the keyboard. All right.
No
No
keyboard
pointer
works.
involved. That's okay. It's my pointer. It should work. All right. So we're going to bounce around in my career and training today. So we'll start out with an outline. I'll give you a little bit of my background. Seth did a good job of doing that for me, so I'll go through it quickly. And then we'll talk about engineered interfaces, both in the context of the development of cell culture platforms for fundamental research and in the translation of these ideas to medical devices. Along the way, I'll introduce you to Charlotte Technologies, my startup company where I'm working. Not mine, I just work there. And I'll tell you about my role in the commercialization pathway for two products that we're making. The first is an intraocular lens, and the second is a wound dressing. And then toward the end of the talk, I'll give you an overview of my research and teaching interests and how they relate to meeting the goals of the Department of Mechanical Engineering here at CSU. And of course, we'll wrap up with acknowledgments. So, like you guys just learned, I started out at the University of Florida. My bachelor's degree is in materials engineering. I focused on polymeric materials. And I stayed at UF for grad school. So I got my PhD in biomedical engineering in Tony Brennan's research group, where I created anti-fouling materials and then modeled the attachment of marine organisms to these materials. After I did that, I was like, well, my degree's in biomedical engineering, but I know stuff about the ocean. Maybe I should go learn about people. So I got a postdoc at Christy Anseth's research group to learn more about how to translate these ideas into the biomedical field. So there I did a postdoc fellowship through NIH and moved into industry after that. So I've been at Charlotte Technologies for about 3 years. I'm the director of product development there and also at a small joint venture company called Clearsight Technologies. But you know, throughout my whole career, I've worked to develop strategies and materials for controlling biological adhesion, and I'm excited to share what I've learned along the way. So we know that materials can be specifically designed to predictably and precisely control biological adhesion. We can design these materials to inhibit or promote adhesion. Everything from surface topography, mechanical properties, surface chemistry, and surface energy all contribute to how organisms like this spore interact with surfaces, and they respond to it by either attaching or not. So we can apply these concepts in a variety of fields, including things as diverse as the marine environment and the human body. So let's start off in the land of my postdoctoral research, where I work to develop cell culture platforms for fundamental research. In this area, it's very important for us to rationally design biomaterials because cells receive signals in vivo from a lot of different sources. They receive biophysical cues from the extracellular matrix, or the proteins that surround the cells. They also get biochemical cues from these proteins. They interact with adhesive ligands, for instance, that are attached to the ECM. And the other sources of signaling are soluble cues that are floating around, and also cell-cell signaling. So when we're designing biomaterials, we sometimes want to recapitulate that and copy exactly what's going on in there, and sometimes we need to simplify that to learn more about how cells interact with their surroundings. One of the advantages we have is that we've been working for a long time in this area. Scientists, engineers, people in medicine have all collaborated to build us this foundational toolbox that we can use that is kind of depicted here in this image. So when I set out to make some cell culture platforms, we started out up here at the top with material selection. We decided to work with a synthetic material, and our hydrogels are based on polyethylene glycol. This is an example of the molecule. It is very hydrophilic, so it absorbs a lot of water, but this structure, when it's cross-linked together, doesn't degrade. So you end up with a material like a contact lens or a piece of Jell-O that absorbs a lot of water and has good elasticity and water content similar to the physiological environment. The other cool thing about polyethylene glycol is that it's a low-Fowler material. So it absorbs very low levels of protein, and that way we can control exactly which proteins we want to present to the cell when we're probing these environments. In my case, we added some adhesive ligands, specifically fibronectin, an adhesive protein, to get the cells to attach better to our surfaces while we were looking at different physical cues we were presenting, including surface topography and surface elasticity. So the cells that I mainly worked with that I'll be presenting to you today are human mesenchymal stem cells. These stem cells are derived from bone marrow, and they're multipotent. That means these cells can differentiate into many different types of tissue, including bone, fat, muscle, and other mesenchymal tissues. The thing we know about these cells is that we can use soluble cues in the media to influence them to either remain progenitor cells, keep proliferating and stay multipotent, or to differentiate down a specific pathway, like turn into a bone cell. The thing that has been discovered more recently is that insoluble matrix cues can also be used to drive this differentiation, and that's something that's being explored that I was really interested in looking at. So we know that cell shape, cell spreading, and cytoskeletal tension all impact lineage commitment, and they all make a significant difference. So these are some examples of cell culture substrates that have been used to examine this phenomenon. A lot of microcontact printing of both proteins and self-assembled monolayers has been done. So pioneering work in Chris Chen's group that's depicted here shows that as these cells are flattened and allowed to spread out, they become bone cells or undergo osteogenesis. If these cells are constrained and stay a little bit rounder, they tend to become fat cells or undergo adipogenesis. Similar work done by Milan Mirshek's group put single cells on these different size islands, and you can see as we move from left to right, we're actually increasing the aspect ratio of the cell, or the ratio of the long axis of the cell to the short axis. When you do that, you actually increase the osteogenic capability. So more of these cells that are stretched out will turn into bone cells. So now we know, well, the shape of the cell is important, aspect ratio is important, and so is radius of curvature. Another type of experiment that's been done is depicted here. This is a polycaprolactone surface with nanopits on it. Dalby's group showed that when these nanopits are arranged in an ordered fashion, the cells that are grown on here retain their ability to differentiate for up to 8 weeks. If you present these same exact nanodots in a disordered array, the cells begin to differentiate into bone cells. So this is really interesting and really impactful work. We could learn a lot from using physical cues to control cells. But a lot of this pioneering work has been done on stiff, static surfaces. And that's not what cells are experiencing in vivo. So our goal was to complement this work and create some surfaces that were softer and more dynamic. So to do that, I chose to use photodegradable hydrogels. This is just a schematic of what a hydrogel might look like after it's been polymerized. These orange dots here represent a photodegradable crosslinker. When you hit this material with light, these photodegradable moieties begin to cleave, and the crosslinks break down and the material begins to degrade. If you do this for a short amount of time, you start to decrease the cross-linking density of the surface of the material and soften the surface of the material. So you can use this technique to control substrate stiffness. If you introduce a photomask and degrade for longer periods of time, you can erode surface topographies. And if you have a really cool laser, you can degrade these surfaces in 3 dimensions and look at how cells respond to these different surfaces in 3D. So my approach was to use a photodegradable macromer that we created in the NSF group. It has our polyethylene glycol chain in the middle, so remember this is really hydrophilic and absorbs a lot of water. Then it's attached to a nitrobenzyl ether-based acrylate. And this is our orange molecule that's photodegradable, and it allows us the functionality to cross-link this network. When we created these hydrogels, we did a chain-initiated copolymerization of this macromer along with a PEG monoacrylate. We initiated the reaction with ammonium persulfate and TMET, and then we end up with a structure that looks similar to this. The other thing we did was incorporate that adhesive ligand that I mentioned earlier, fibronectin, into these surfaces to help promote cell adhesion with one specific signal. So I'm really interested in topography. That's kind of the underlying theme of my talk. And we wanted to create some micro-topographies in the surfaces. One really cool advantage of using a photodegradable hydrogel is that you can swell these surfaces prior to patterning. So you end up with feature sizes that are more similar to your photomask than you would if you were, for example, casting the hydrogel material and then swelling into equilibrium. So we start out with a hydrogel that's labeled here in red. We incorporated a fluorophore into our materials, so you'll see that the actual photos of micrographs of our materials are all red as well. This is an example of a chromon quartz photomask that we placed on top of the material. We expose it to UV light, and then we come out with a topography. So the areas that are masked and not exposed to light remain, and the areas that are exposed to light degrade away. These are Z-stack confocal images that might be a little hard to see, that just show you qualitatively that as we increase the exposure time at a set power of our irradiation, 10 milliwatts per centimeter squared, we increase feature depth, and that was to be expected. So when we quantify that using contact profilometry, you see as the irradiation time increases, our feature height increases almost linearly. Our goal was to use these cell culture platforms to control the cell shape of an entire population of cells. So we want the cells to be able to spread across the surface and not be blocked or prohibited by very tall topographies. So the work I'll show you today was done at this 1 micron height with a 250-second exposure time. So we started out just looking at how these stem cells respond to static topographies and to see if we could stretch out or change the aspect ratio or somehow change the shape and spreading of these cells. So our MSCs were derived from bone marrow. We grew them on traditional polystyrene plates for no more than 3 passages and then seeded them onto the surfaces at fairly low densities of about 6,000 cells per centimeter squared. When you look at our topographies, they were intentionally designed to start out with an isotropic aspect ratio of the features. Thank you.
I don't know, that do it, but off.
Yeah, you just have to turn it back on after a while.
Okay.
Don't fall asleep. So we increase the aspect ratio of these features from something that's very isotropic to something that's very anisotropic in this channels pattern. And then we stain the cells for different components that are inside cells. So the red here is actin. It's a protein that makes up the cytoskeleton in the cells. The green labeled here is vinculin. That's a protein that is in the focal adhesion sites of cells. Those interact with the surface. And then the blue is the cell nuclei. So you can see qualitatively, as we increase aspect ratio of our features, we're increasing elongation and orientation of our cells. So to quantify this, I wrote some fancy MATLAB code. I was really proud of myself. So we started out by labeling our cells with Cell Tracker. It's a cell membrane dye, and then taking fluorescent images. We took those images, adjusted the brightness, contrast, and thresholded. So now we end up with a white cell and a black background. We also removed cell aggregates. And then the software fit an ellipse to each cell. So then we could start to measure different features of our cells. So like I mentioned, aspect ratio is important. So we measured the aspect ratio of our cell, ratio of A to B here. And then we also measured the angle of orientation, the angle between the long axis of the cell and the direction of the topographic features. So this is another cool MATLAB output. All of these ovals represent a single cell that we measured on top of our surfaces. So we did 3 replicates of each surface and took many images per replicate. And again, you can see this qualitative pattern emerging where as you increase aspect ratio, you're significantly increasing elongation and orientation of these cells. When you quantify that, you see that we end up with sort of 3 groups that we can fit these cells into. Aspect ratios that are the same as smooth, ones that are significantly higher, and then on the channels, significantly higher than that. So now we have sort of 3 groups to use when we're influencing our cells. When you look at the percentage of aligned cells, we don't increase cellular alignment until we get to really high aspect ratios of 8:1, or our channels, which we kind of called infinity compared to the size of the cell. So, in addition to being able to control an entire cell population instead of just one cell at a time, we have this other really cool aspect of our substrates, which is a really neat advantage. We can dynamically pattern them using cytocompatible light at 365 nanometers, so we can change the surface while the cells are on it. This is an example of dynamic patterning. We start out with a smooth hydrogel, we introduce a channels topography, and then we cut those channels into squares. So while this is a reductive process, you'll have kind of a limited number of times you can do this, and there's still some really interesting things we can figure out. So here's an example of some cells on our surfaces and looking at in situ control of cell morphology. We again labeled with cell tracker and seeded these cells onto smooth surfaces and imaged them using live imaging techniques. After 24 hours, we introduced a channels pattern, let the cells reorient themselves, and then imaged again. You can see now they're stretching out on the surface in response to the topography. After that, we cut those channels up into these squares patterns, made a more isotropic pattern, and the cells returned to their original shape. So when you quantify that, you can see that there's an increase in our average aspect ratio when we dynamically pattern, and also an increase in alignment and then return. So this is a really powerful tool. The work that I told you about in the beginning, where they were increasing osteogenesis using these different aspect ratios, induced cellular aspect ratios of 4:1 or 8:1 to really increase osteogenesis. Those are achievable using our substrates, and I believe the next step of this is to really understand better how we can change cell function and influence it. And I just wanted to throw out an idea for translation, because that's where the next part of this talk is heading. I believe that one way you can utilize these surfaces is in stem cell therapies or in tissue engineering, where you might isolate stem cells from a patient, and then you only get a few of them, right? So you have to expand these cells. You could do that on a surface that helps to maintain multipotency, or the ability to differentiate. And then while these cells are on the surface, in situ, you could change the topography underneath them and tell them to differentiate into a specific lineage. Once you have those cells that may be bone cells, you could implant them again on a tissue engineered scaffold and help to reduce the time that it takes from taking cells from a patient to getting them back in. So now that we have all these cool materials ideas, how do we translate them? I'd like to share with you a little bit about how we do that at Sharklet Technologies, and I'll go over 2 different projects with you. So Sharklet Technologies, our vision is to invent non-toxic topography-based medical devices to help improve human health. We were founded in 2007, and we're headquartered in Aurora. We believe that Sharklet is the first surface texture that's been engineered to specifically control biological adhesion and marketed that way. It was started, the idea for Sharklet came from my PhD research group. So our first patents were licensed from UF, and we have a lot of pending patents that are both filed by Sharklet and licensed from the university. The cool thing about our material that you'll see throughout the talk is that Sharklet is a platform technology, so we can apply the ideas and concepts of using topography in many different fields. Here are a few examples of product applications where I have done a lot of work since joining Sharklet. The first is, our first set is medical devices, but as you make a medical device, you have to go through the regulatory process, which could take up to 10 years and cost a lot of money. So the secondary objective that we have is to make surface protection products, products that keep bacteria from attaching to your surfaces or transferring. You would think of high-touch surfaces in hospitals, something like your cell phone case that you don't want to get dirty. So this cell phone case is a feat in manufacturing for us. That has a microtopography of it on it with a resolution of 2 microns. So I've been involved with making that manufacturing process work. That was injection molded. I've also worked on several medical devices. The first is an endotracheal tube that we use for airway management when someone's on a ventilator. The next is an intraocular lens that we'll apply in ophthalmology. And the final product is a wound dressing. So I'll start by telling you a little bit about how Sharklet works. Sharklet is bio-inspired. And we get this inspiration from the skin of sharks. So you can see on this whale, he's covered with biofouling. That's what we like to call the plants, animals, and organisms and their byproducts that stick to things in the ocean. You're probably familiar with barnacles stuck to a whale's nose, but there are bigger implications. Biofouling increases the cost of shipping goods. Over 80% of the goods that are sold in the entire world are shipped at some point. It also increases the the amount of drag that occurs on naval ships. So it's a national security issue. We want to increase our fuel efficiency, decrease the amount of time that we're dry docked and cleaning ships. And biofouling also occurs in heat exchangers for power plants and other areas like that. So when my PhD research group was trying to address this problem of biofouling in the ocean, we looked to sharks for some inspiration, because you can see they live in the same environment and there's nothing stuck to their skin. So when you have your advisor in Florida say, "Go catch me a shark," somebody does it. So one of our graduate students caught a shark. We took some dental impression material, stuck it to the side of the shark, and pulled it away and looked at it under a microscope. When you do that, the pictures aren't as pretty as this SEM, but you see that shark skin has a really distinctive topography. So it's made of these dermal denticles that are shaped like a diamond with raised ridges. We believe that this is really important in the fouling defense mechanism of the shark. So we created this bio-inspired topography that we call sharklet. It still has this diamond pattern. We have raised ridges, but it's on a smaller size scale. So these bars are 2 microns wide and 3 microns tall. So this was originally tested against a lot of marine fouling organisms. And this is the culmination of my PhD work. It's this thing. So,
All the students appreciate that.
it's right here. So how does it work? Well, when you're in the marine environment or in a biological environment, your surfaces are usually in contact with water or some other type of fluid. They're wetted. So sharklet is based on wetting theory, and how we model it and interpret it is based on wetting theory. So I'm going to tell you a little bit about wetting theory. Is anybody excited about wetting theory? Yeah. Yes. All right. So when a liquid comes into contact with a solid, the droplet that forms creates a contact angle. And that contact angle can be explained by Young's equation, where the contact angle is equal to some relation of these different surface energies that exist, or surface tensions. And this all changes when you add a microtopography to a surface. So when you have a micropattern surface, you can wet in 2 different regimes. The first is Wenzel wetting, where the apparent contact angle, the contact angle you measure on the surface without knowing that it has topography, is equal to the contact angle on a smooth surface made of the same chemistry, multiplied by R, which is the Wenzel roughness factor. So R is the ratio of the projected planar surface area that you create with your topography to the actual surface area. And when you use this R number, you can predict what your contact angle will be based on what pattern you put on the surface. The other regime that you can wet in is the Cassie-Baxter regime, and that happens in 2 different scenarios. The first is air pockets, where your bubble actually sits on top of the microtopography and no air gets underneath. And then the second is wicking, where your bubble actually wicks out at the leading edge. So I pointed out R in the Wenzel roughness equation. The thing that we are really interested in is this piece of S in the Cassie-Baxter equation. It helps us to describe the area that is not in contact with water or liquid underneath the droplet. So my PhD work, we knew we had this surface sharklet that keeps things from sticking to it, but why does it work? And why does it work better than channels, or why does it work better than pillars? We needed some way to mathematically model this, or to help us design things that maybe would work even better than sharklet. So we developed this empirical model called the Engineered Roughness Index. And we used R from Wenzel wetting and this piece of S from Cassie-Baxter wetting to help us mathematically describe our surface and what our features looked like and the area of those features. And then I helped add in a few other components that I thought were really important. So we're testing a lot of different types of organisms, right? We're testing a spore that can swim, and it's about 5 microns in diameter. We're also testing bacteria, which are much smaller. Sometimes they have a flagella and sometimes they don't, which means sometimes they swim and sometimes they don't. And we're testing these on lots of different types of surface chemistries. So first we added in the Reynolds number of the organism to help try to describe the size, shape, and motion of the organism through the fluid. And that way we're hoping we can model our surface topography and how it interacts with lots of different organisms. And then we also added in a surface energy term to help describe the surface chemistry. So it's more of a chemistry-based energy term than a topography-based term. So once I did a whole bunch of experiments and modeling, came up with this line based on those terms. So this is the normalized attachment to our surfaces. And we have a pretty good correlation coefficient here of 0.8. So these are experiments ranging over 3 years with 3 different organisms from different phylogenetic groups. And we're able to model this attachment. So now this equation and these parameters are used when we try to identify the correct surfaces or maybe invent new surfaces at Sharklet to prevent biological adhesion.
I have a real quick question about that. So more adhesion is going down, or you said that, what is on the y-axis?
Yeah, so we're decreasing adhesion, so this is less adhesion.
Got it. I just want to make sure I'm reading that right.
Yeah, so higher roughness value based on our model is lower adhesion. So even though our surfaces were originally designed to keep organisms like algae in the ocean from sticking to them, we quickly learned that they also keep human pathogens like E. coli and Staph aureus that cause infections in hospitals from sticking to the surfaces as well. So that's how we turned into a medical device company. But when I joined Sharklet, I have experience in cell culture and tissue engineering. I don't know anything about microbiology, and so I wanted to shake this up a little bit. So using our similar toolbox to the one I showed you before, but basing this all on our Sharklet surface, we can change the way we interact with our organisms. So instead of inhibiting biological adhesion, we can promote or direct it, and that's where the focus of my work at Sharklet has been. So again, we can change our chemistry. We can change aspects of our topography, keeping the same Sharklet base. We can make the features larger. We can change the orientation. We can make a radial design instead of a square design, things like that. And then also we can control the mechanical properties of our surfaces. So with those things in mind, I'm going to introduce you to two projects that don't have anything to do with bacteria, and these are the ones that made me the happiest. So the first is we're working on an intraocular lens. So cataract is a leading cause of visual impairment in the world. And when you have a cataract, the treatment is to remove your natural lens and replace it with an artificial one. The current artificial lenses look like this. There's an optic in the middle, and they have these two arms that come out and hold it in place. They're called haptics. In 50% of cataract surgeries, up to 50% of cataract surgeries, there's a complication that occurs called PCO, or posterior capsule opacification. What happens is the lens epithelial cells that are left behind in the capsule begin to migrate and try to kind of heal the wound that's been created by removing the natural lens, and they end up blocking your vision a second time. So you can see here, all of these cells that are behind the optic are starting to interfere with this patient's vision, and that's a problem. The only treatment for this is called a YAG laser capsulotomy. They actually take a laser, shine it into your eye, and ablate those cells. But this ends up being a pretty expensive procedure, and a lot of them get done. So 600,000 of them happened in 2012, and it cost over $250 million just to Medicare alone. So this is something we're trying to address because we see a need to address this problem now as our population is growing older and having more comorbidities such as diabetes. These numbers are skyrocketing. So the first step to addressing this problem, we thought, was using topography to control where these cells are going. So we selected and created 3 different microtopographies to test, and then this is an example of our benchtop test. So usually when people work in the wound healing space, they do what's called a scratch wound assay. They grow cells in a petri dish until the cells are all touching each other or confluent, and then they actually take a pipette tip and they just scrape down the middle to create an artificial wound. Once you do that, you can watch the cells migrate into that new space, and you can give them different cues to encourage or discourage that. We can't do that on our surfaces because we would destroy our microtopographies. So we made this modified assay where we put down our microtopographies, we put a silicone patch on top, and then we seed the cells. When you do that, you're effectively blocking cells from landing in the center here. So you let the cells grow to confluence, you remove the patch, and now you have a space where you can watch cell migration coming from the edges. So the images that I'll show you are spanning the edges, so you'll see some of the confluent cells and then the cells moving into the center. For this assay, we used lens epithelial cells derived from humans, and we stained them with that same cell tracker dye I used in my postdoc, and we looked at migration after 7 days. So you can see on these surfaces that the cells begin to migrate in from the edges and actually start to close in the wound on the smooth topography. On a few different surfaces that we tested, we saw some reductions in this, and then on the surface we actually designed to block cell migration and we thought would work best, did, so that's awesome. Our theory's matching up with our experiment. We see an 80% reduction on this larger micropattern. So instead of being 2 microns wide, those features are 10 microns wide with 5 microns spacing in between. So this was really exciting and encouraging for us, and we started down the pathway of prototyping what our lens will actually look like so we could test it in vivo. So this is a first generation idea of what our lens would look like. It's completely different from what is currently on the market. We have an optic in the center. Instead of those haptic arms, which are actually typically a nucleation point for that cell migration problem that causes PCO, we've created a ring haptic. So now this ring stays in place to keep the capsular bag open, and in between the haptic and the ring is a sharklet pattern membrane, where the sharklet pattern is on the posterior side to contact those cells and block them from migrating into the optic space. So that's something that's really complicated to prototype and manufacture. So to test this out, we created what we call a protective membrane. And what we did was we made this membrane, we made the ring haptic, but we didn't put an optic in the middle, because who cares if the rabbits can see? And then we actually implanted an IOL inside our ring. So the haptics are not interacting with the bag, and they're not causing a nucleus for cell migration because they're inside our membrane. And we could create this in 2 pieces, and we implanted it into a rabbit model that's typically used to test PCO. So I don't know if you caught me, I was being sneaky earlier. When I showed you our migration assay, we had aligned the features perpendicular to the direction of migration. We did that on purpose. Because when the features are perpendicular to the direction of migration, they block migration. If they're parallel to the direction of migration, you can actually enhance migration. So in order to address that in a round environment, we had to create a new pattern. Because we need more mechanical engineering expertise on our team, we did this quick and dirty. And so we made a checkerboard pattern where these are about 500 microns wide, or across. And so the cells can migrate for about 500 microns in any direction before they bump into a topography that's turned in the way that should block them. We've since then started working on a radial design where we actually had to add some arc to the features to create what we needed. So when we test this in a rabbit model for PCO, we get really, really good results. So this is an example of an eye that has been imaged in a slit lamp exam. So they shine a light in and take a picture. You can see all of these are all cells that have migrated in and blocked vision. This is an IOL that's actually on the market right now, sold by a competitor. Here is the same IOL, but inside our protective membrane. You can kind of see the checkerboard pattern up in the corner. When we scored PCO histologically, we saw a 70% reduction in PCO on our design versus a regular IOL. And then we showed all of these images to 3 different ophthalmologists, and we asked them, would you treat this with a laser or not? And 100% of them said no on all of our eyes that had our prototype in them. So that was really exciting. This work was originally funded by a phase 1 SBIR grant that I worked on as a consultant. Since then, I led the charge in doing all of the in vitro and in vivo work, although I'm not the PI on the grant. And I helped us to submit a phase 2 that we just got awarded and have started working on. So the phase 2 grant is about $1.8 million for us to prototype and start to move toward clinical studies. So here's a really fun part of our project. When you put these in the eye, the intraocular lenses have to be folded and inserted through an incision that is 2.8 millimeters wide or less. So we need to create an injector that will fold the lens, keep it in the proper orientation, and put it into the eye. To do this, we've actually been working with a senior design team in BME at CSU, and they're designing our new injector. I also made them do a lot of paperwork. So they're learning a lot about the process that we work under using a quality system. So the first thing we did was we started interviewing physicians. We went and visited an ophthalmologist and watched them insert normal lenses with an injector. And